Hey everyone, so this is D again. Um, we went over cells as cellular metabolism today, so we'll go over that first, and then after that we'll go over tissues and the basics of the integumentary system. We didn't really touch about it much today, but we will go over it a little bit in the second video. In the first one, we're gonna do cells and cellular metabolism. So let's get started. So I drew here for you a basic animal cell or eukaryotic cell versus the bacterial cell that we have here. So based on the lecture, we worked our way inside and somewhat outside as well as back inside the cell. So to make it a little bit more interesting and more sequential, we work our way from outside all the way deep inside into the cell and then compare it to the bacterial cell or the prokaryotic cell that he was talking about later on in lecture. So as you can imagine, the human cell has the Golgi apparatus. It has the smooth and rough endoplasmic reticulum, lysosomes, peroxisomes. Of course, it has the mitochondria, or the powerhouse of the cell, the Golgi apparatus, smooth ER, rough ER, kind of like the post office of the cell. They kind of ship, package, and use vesicles to operate that. Lysosomes obviously help with digestion of biological and biochemical particles inside the cell, and also help with apoptosis, as we already know about. We also have the microtubules. We have the microfilaments. We have the nucleus and nucleolus. We have the vacuole. So I made an error in my first video, but I corrected it in the comments on the Facebook page. We also have, let's see here. So we have the Golgi, smooth, rough ER, lysosomes, peroxisome, mitochondria, microtubules, microfilaments, nucleus and nucleolus, vacuole, cytoplasm, and the cell's cytosol, which is the fluid itself. And of course, we have free-floating ribosomes and bounded ones for the rough endoplasmic reticulum. So as far as that, let's get into it. So we worked from the cell membrane, which is this outer layer out here. We said that it's a phospholipid bilayer, so it's has a side that's kind of, as he said before, two-faced in a way, or schizoid, due to the fact that you have the hydrophobic tails that are on the inside facing each other. They look something like this. So that's what that looks like. So that's basically what that cell membrane is being made out of. Here, okay? So the takeaway from that is, A, it allows for a selective permeability, or some things can get through while some things can't, or not as easily. The other thing is, hydrophobic is on the inside, hydrophilic is on the outside. Hydrophilic, hydrophobic. Alright? Next thing he talked about was the function of the cell membrane insofar as getting things in and out. So the way that things get in and out is through phagocytosis, which is eating by the cell. So phage, eat, cyto, cell, osis, the way of. That's it. The way that the cell eats. Penocytosis, the way that the cell drinks or takes things into itself. Exocytosis, exo meaning pushing out, where things go out of the cell, either by vesicles or proteins or channels, whatever it may be. Usually requires some form of ATP and some protein or some vesicle, usually provided by either the Golgi or by the peroxisomes, okay? So that's important. Then you have endocytosis, which phagocytosis and penocytosis fall under that umbrella term, which really means you're taking things into the cell, or things that are already in the cell, you're bringing them further into the cell, closer to where they need to be organelle-wise. Okay, the next thing from there is going to be the protection factor. So the proteins, they're integral proteins, proteins that are embedded within the cell membrane. So they're embedded kind of like that. So that's, imagine my pen being an integral protein. It's integrated within, it can't move, along the bilayer. Then you have proteins that are able to facilitate how things get into and out of the cell, but are not integral. Those usually are globular proteins, for instance. And before, he talked about globular or G proteins being things that bind to things in order to get the primary messenger, which is maybe epinephrine or butyrol, for instance, binding to a receptor. So I'll show you what that looks like. So on the inside of a cell, you have a receptor, and the E here is going to stand for epinephrine, and the little dots are going to be the secondary messenger. So as you can see here, you have cell membrane, phospholipid bilayer, integral protein somewhere within the phospholipid bilayer. Close-up shot of that is the receptor, which is kind of like the lock. Epinephrine, which is the key for that particular lock. I don't like to really call it a lock and key method because that's usually reserved for enzymes and substrates. So that's usually talking about cellular metabolism. So things like cofactors, as well as allosteric sites, allosteric inhibitors, all those fancy terms that just really mean places where that substrates such as other minerals can bind to onto an enzyme. But for all intents and purposes, right here, epinephrine is nicely fitting into this protein, and then it's sending out the secondary messengers for inside of the cell. So you got that much, okay? So that's what he went over for the first part. I hope that's pretty straightforward. What he talked about as well in terms of proteins is the gated channels versus a pore channel versus the different ways that materials get into and out of the cell, which is usually the osmosis, diffusion, and active transport. So the main difference between a gated and a pour is that for the gated, it's about opening and closing and accumulation of charges to force these doors, quote unquote doors, open and close. Next thing you have is pour channels. So the pour channels are usually cylindrical. So you can think of this like, I'll actually draw it for you. You can think of it as if you're inside of the cell, and you have the phospholipid bilayer as we spoke of before. So this big guy right here, the cylindrical protein that's in between the phospholipid bilayer, this is representative of the pore channel. So this one is cylindrical. Usually it likes aqueous solutions, so that's reason why that one side of it is open and it's free flowing, allowing ions to travel freely inside and outside of the cell. But unlike gated channels, it's far more selective. It might sound counterintuitive, but it's specific to certain ions and to certain materials that it will let in and out. So it's called a fixed pore channel in the sense that it's limited by the ions that it can accept or refuse. While gated channels, which need energy and need certain ion gradients, may allow more than one type of ion to freely pass through, depending on whether it passes the threshold or passes the level necessary to open or close its gates. Make sense? Alright, cool. So now, moving a little bit downward into the phospholipid bilayer, you're gonna have can't draw very well, but you're gonna have things like cholesterol inside of the phospholipid bilayer. So what cholesterol is gonna do for you, which is important for our job at least, is that it helps with heat and cold. So if you stand in front of an AC with your arm like this for about 5 minutes, you might feel a little bit breezy. It's like, that feels pretty nice. You stand there for about 2 hours, you might feel a little tingly feeling, but your skin is not gonna dry out because of the cholesterol, because of its fattiness. To be more specific, because of the glycerol backbone that you read in the textbook and how it binds, and because usually cholesterol is a very long molecule, it's not readily broken down by changes in heat. However, what's the most important thing about cholesterol is that you see these rings here? That's what keeps it from breaking down. The phospholipid bilayer has the two little tails, has the head, but nothing much else. So it's pretty easy to get through this without the proteins. That's why the cholesterol is here as well. Cholesterol can actually block the phospholipid bilayer too, with the aid of proteins, of course, either from inside or outside of the cell, depending on the needs. But because of its ring structure, just like the pyrimidines and the purines in terms of the nucleotide bases, it's a very rigid molecule. So because of its rigidity, it doesn't break down as easily. A comparison might be olive oil. So if our cholesterol was basically coming from olive oil and was within our phospholipid bilayer, instead of being blocky, it's going to look something like this. It's going to look very thin. But because of that thinness and obscure shape, it's more longitudinal than it is lateral, as well as having some 3D portions to it. It breaks easily. So if you stand out in the cold, you'll freeze very easily. If you are in a desert, you're most likely gonna sizzle very quickly as well. So that's why for the cell membrane, we have cholesterol, proteins, and a phospholipid bilayer. Alright, moving on. So the important things that we have to worry about in terms of the receptiveness of the cell membrane is overdosing on medications or drugs. For instance, albuterol, if you take it too much over time, you have a downgrade or downregulation of the receptors. Beta 2, I believe, receptors it is. Yes, beta 2. Because it also affects the GI tract, the uterine wall, the vasculature of the body, as well as the airway. Epinephrine, however, is going to bind. It's going to be the primary messenger, then it's going to have a receptor, then you're going to have a secondary messenger from inside the cell going out. Then from there, the Golgi apparatus will kind of help to downregulate by encapsulating the receptors in vesicles. Alright, so moving deeper inside the cell now, we have where protein synthesis by ribosomes or amino acids, which are acid having nitrogen or having R groups, which is in your textbook, for different functions, of course, and essentially it's the building block of protein. So that's how it's easy to remember. Amino, building block. And the reason why it's called amino is because of the amine group, the AMINE group. So there's a list, there's like hydroxyl, carboxyl, all of that good stuff, superhydro, etc. But amino gets its name from amine. So for protein synthesis, first we look towards the nucleus. So for the nucleus, we get information. The way that we get that information is via first translation. The reason why it's translated first before it's transcribed or transcription is due to the fact that, pretend this is your nucleus. It's not going to look this pretty in a real cell, but whatever. The DNA is actually super coiled. You don't have to worry about what super coiled means, but let's just say that it's something like this. Okay? It's super coiled, it's compacted upon itself, and it has methyl groups, and you don't want to be heavy methylated because, you know, it changes the nature of the DNA, meaning that it becomes more acidic. It's tightly packed in here, so in order to get it out, first just like the proteins with the cell membrane, it needs a messenger. So guess what? It has messenger RNA that can exist between the nucleus and the nucleolus, which is right outside of the nucleus. So, of course, there's a protein that allows the release of the mRNA from the nucleolus. And then what happens is you have our friend Mr. Ribosome, free-floating ribosomes, of course, who comes along with the 3 spots filled in, the larger portion, which you don't need to know, and the smaller portion, the larger subunit and the smaller subunit. And it comes to transcribe the information. So, for DNA, as you know, it has gap, which is guanine and adenine, which are the purines. Then you have uracil, cytosine, and what's the last T one? I think it's not threonine. It's another nucleotide base. You can look it up as well. But you would have the pyrimidines and the purines. So you have cytosine, adenine, guanine. Can't get the T. I don't know. I already don't know. I can't remember the last name. Cytosine, adenine, guanine, and some tyrosine or something like that. But that's not the point. The point is that the body, especially the body of the DNA cell, accepts T, but RNA does not. So it actually prefers uracil. So that's where it's translated. Then it's transcribed from just the nucleotide bases. Nucleotide bases are 3, come in 3s. And that's how the ribosome is able to make protein, because there's a whole chart in your book that has the different amino acids, such as like leucine, valine, tyrosine, taurine, glutamine, etc., etc., etc. And they're made by the 3 nucleotide bases, or in more technical terms, a codon. So you have on the ribosome a place where you start and a place where you end. So for all intents and purposes, let's say this first one here is start and here is stop. So it'll transcribe each and every nucleotide base, and then it'll break off, and then you'll have a transcription RNA or tRNA that then flips it. So now it goes back to being the amino acids in the pure state before they're packaged. So originally the amino acids have to have, which is not important for this class of course, it has to have subquarter packages, not just like free-floating amino acids. It's packaged by a poly tail and a 5A cap. So it has a tail and a head, just like the phospholipid bilayer. And that's how it doesn't go haywire from translation to transcription. Then you have transcription RNA, which then binds the amino acids together to make a protein. Then another larger protein comes and bends it and twists it so that it forms the alpha helix, which is the first structure, the beta plated sheet, the tertiary structure, and the quaternary structure, which is just basically, as Gary said before, proteins have functions based on their structures. So you'll have one that looks like a sheet of paper. You'll have one that just looks like a helix. You'll have one that looks more pronounced. So alpha, beta, tertiary. Quaternary looks a little bit more crazy, but you get the idea. Okay, so that's for that. Going further into the cell now, you have lysosomes, which is digesting or breaking down the molecules that are either just garbage, toxic to the cell, or what the cell actually doesn't need anymore. So like old proteins, old organelles get chewed up by the lysosome. So it's the garbage man of the cell. The mitochondria, which is most important, microtubules, microfilaments, cytoplasm itself, and the Golgi are a lot more important than the others. So that's what I'll talk about right now. So for the smooth and rough endoplasmic reticulum, just know that one is studied, one is not with ribosomes, and you can read more about it in the textbook. The cytoplasm itself produces cytosol, or liquid. It's similar to like how blood has plasma and it has the serum part of it. So just think about it that way. It's the environment in which the organelles live in. And to be honest, it's slightly more alkaline than even our blood is. Particles with different charges can also exist within the cell, within the cytosol, which is like the more liquidy portion, than the jelly portion, the cytoplasm. Similar to how in the skin, and the mucus, you have the thick portion and the thin portion that he talked about in class. Okay? So cytosol is easier to go through than cytoplasm. Cytoplasm you'll find more at the thickest around that cell membrane. So that's how you can remember that. But cytoplasm and cytosol are usually mixed together within animal cells. So next thing from there is going to be the mitochondria. So for energy production, most important thing is that, based on our lecture, aspirin changes in electrons or charges, and even the presence of more than extraneous ATP stops the Krebs cycle, or stops the TCA cycle, or stops the citric acid cycle. Whatever you want to call it, is a cycle where you produce 36 ATP at the end of the day. You also want to understand that cyanide interrupts, which is a poison, interrupts the electron transport chain. The reason it does that is due to the fact that oxygen goes bye-bye when cyanide is there. So you'll have, obviously, the presentation of a patient that's flushed and hypoperfusing. From there, you want to understand that energy production can occur two ways, with or without oxygen, except for in the cyanide case where that's more based on poisons. But I'm talking about just normal energy production with or without oxygen. So you have one, which is the glycolysis, the breaking down of glucose, lysis to lyse, lysosome. See how it all makes sense? And glucose gets broken down usually without the presence of O2. Without the presence of O2. Byproduct of this is lactic acid. This part of the process is also called lactic acid fermentation because if you have a dried up piece of fruit, it ferments in the sun, so you can think about it the same way. Or for you gym buffs out there, you can think of the acid building up in your muscles, so that's why you feel major pumps.